Welcome to our channel Bio by Gulab Sir for Wisdom Question Center. As a part of series of lectures, today's topic is Law of Dominance. In last class, we had learned about Mendel's hybridization experiment and his contribution towards genetics, as he is honored as a father of genetics. He was the first scientist and a great human being to introduce genetics with statistical analysis, mathematical and logical explanation. It was really new for the world that biology with logical, statistical and mathematical explanation. Therefore, by the time when he produced his experiment and produced his data with proof that nobody could understand his work, and it remained hidden for 40 to 50 years, that he was published his paper in 1857-64 and it was hidden up to 1905. But the rediscoverer of Mendel, Hugo de Vries, Correns and Seremar, that they separately work on Mendel's hybridization experiment and came to the same conclusion that Mendel has made. And thereafter that Mendel work was recognized by whole world. So as a part of his work, that he has carried out his experiment by having 3 steps: pure line selection, hybridization experiment and formulation of laws. So as a part of syllabus, that law of dominance is the first law that he put forth. To understand that pure line selection, in short, that when he selected these 7 pair of contrasting characters, that he could produce, and he was very intelligent enough to produce all these plant for pure trait, that growing tall plant for many generations, observing that tall plant produce only tall and dark plant seed when sown and grown produce only dark for many generations. That is called pure line selection. Because it was very important to carry out hybridization experiment to have that plant with purity for that character. That means tall plant produce tall, dark plant produce dark, so that with that cross hybridization experiment, what the conclusion after that hybridization seed obtained is sown and will produce. On the basis of that, that experiment were based. So let us understand first law of dominance. As we had discussed about this 7 pair of contrasting character and why he did selected pea plant for his experiment, that we had discussed. And this is what data given on NEET. Now to understand, we have to begin with our own way of explanation so that you will understand. So here, first law of dominance. Law of dominance. He carried out hybridization experiment on pea plant by having first experiment with character, that one plant pure for its character, that is tall plant, cross with dwarf plant. These were the parent that he selected. These were the parents that he selected for his experiment, that tall plant and dwarf plant. Now, by the time when he discovered these laws, genes were not known as by name gene. He called them as a factor. So he considered that factors that control the tallness and factors that control the dwarfness could be as a genotype. Genotype are the setup of gene that control this character. So tall plant will have genotypes capital T, and dwarf plants have genotype small t. What does that capital T and small t is to denote as a gene? Capital T is a dominant, that what trait is controlled by capital T means a gene which expresses its expression, and small t which could not produce any protein, therefore do not express in presence of this capital T. Now as a meiosis, when any cell undergo meiosis to produce the gamete, it what produce gamete with haploid number of chromosome. Since these are the capital T say as 14 number of chromosome in T plant, P plant, here this undergo meiosis, that is gametogenesis, producing gamete. with capital T, that is 50%. Out of 2, 1 only will get into the gamete here with small t. So this is haploid, that with 7 number of chromosome. So this was diploid, and now these gamete produced are haploid. So this is what to understand. So these are the gametes. Now when these plants cross, what does it mean? That pollen grain from one plant to the stigma of the another plant is transferred. And as we learn what is artificial hybridization, in that we have emasculation, bagging, transformation of pollen grain on the stigma, rebagging, and waiting for result. The same way, same experiment is carried out by Mendel. So here these are the gametes. Now crossing, that is called hybridization or fertilization. Crossing can be called as a step fertilization, where that pollen grain from one to the stigma of the another is transferred, and when the seed is produced, that first filial generation, which obtain having these two sets of genes together, this is what first filial generation, F1 generation. Now he did observe that only one character of the trait, contrasting character of the trait is height, contrasting characters are tall and dwarf, only one that is observed in F1 generation. Therefore 100% plants were tall. So you got surprised that where the dwarf has gone. When tall was crossed with dwarf, only tall plants appear in F1 generation. Where the dwarf plant has, why that dwarf plant not appear in F1 generation? To understand that, he decided to do the selfing. So selfing does means that transformation of pollen grain from the anther to the stigma of the same flower. Say this is the ovary, these are the anthers, and this is a flower. For selfing, transformation of pollen grain from anther to the stigma of the same flower, that is called selfing. So here when he made selfing, that he had Punnett square. This Punnett square is like this. So because of heterozygous condition, can produce two types of gamete as capital T and small t for the male, and female capital T, small t. So when Punnett square is made, see this is Punnett square. Punnett square. So here Punnett square help to know that when this egg cell is fertilized by pollen grain having capital T, will lead to produce a genotype with capital T, a tall plant. When this is crossed with this one, will produce a zygote with capital T, small t, this one with capital T, small t, and this egg which fertilized by pollen grain having small t will have small t, small t. That means when he did observe after crossing that out of 1000 plant, around 750 plants were tall. That means 3/4 of the plant were tall and 1/4 of the plant were dwarf. So here this tall and dwarf. When he did observe that in F1 generation 100% plants were tall, and in F2 generation the ratio was tall to dwarf, as a phenotypic ratio was 3:1. This is phenotypic ratio. What is this? Phenotypic ratio. 75% tall, 25% dwarf. When he saw that few plants, that is about 25% of the plants are dwarf, that means the gene which control the dwarfness was present in F1 generation but could not express. That means that gene which do not express in presence of its allele. What are allele? Gene that control a trait. That contrasting pair of gene, capital T for tallness and small t for dwarfness, are called as allele. So small t as a allele could not express in F1 generation in presence of its allele, capital T, is known as recessive gene, and capital T which express in presence of its allele small t is known as dominant gene. That means a tallness is controlled by capital T. Unless and until these dwarf factors, that is small t come together in homozygous condition, that dwarf plant could not produce, as it is seen in these two conditions. That these are the heterozygous condition. What does it mean? One gene, that factor is capital T, another factor is small t. This condition is called heterozygous condition. And capital T and small t, these two are called homozygous, since both the factors are same, that they control tall, control dwarf. But here one control tallness, another control dwarfness, although small t, that is dwarfness, could not express in presence of that capital T, that means in heterozygous condition. So plant with genotype capital T small t express only tallness, and therefore the gene which express in F1 generation in presence of its allele small t is called dominant gene, and the phenomenon is known as law of dominance. Same experiment he made for other 6 pair of contrasting character. That 6 pair of contrasting characters were left this tall, that position of the flower, color of the flower, color of the pod, shape of the pod, color of the seed and shape of the seed. In every condition, when he cross, that always that factor which control a trait, that capital R, capital Y, capital I, capital Z, capital P, capital A, that they express in F1 generation, and in F2 generation both the character were seen, or factors were seen in the ratio, or trait seen in the ratio 3:1 for all the experiment. So this was a logical experiment, this was a mathematical explanation, this was a statistical data which could produce by the Mendel all the traits, and therefore he came to conclusion that the factor which express in F1 generation is called dominant factor, and the phenomenon is known as law of dominance. As when he cross a pure tall tree plant with pure dwarf tree plant, only tall plants are produced in F1 generation because of that factors which express in F1 generation in presence of his allele small t. Therefore, he called that capital T, that which express in F1 generation in presence of his allele, called as a dominant factor, as a dominant gene. And phenomenon what he explained for all other 6 traits also, he called as a law of dominance. I hope you did understand what is law of dominance. Question can be asked in board that what is law of dominance, explain it with suitable example or with specific cross. Also can be explained, ask what is monohybrid cross, explain it. What is monohybrid cross? When he consider only one trait for his experiment. So monohybrid cross. When only one trait is considered for hybridization experiment, is known as, and that cross is known as monohybrid cross. And yes, with the help of this monohybrid cross, he could explain law of dominance. What is law of dominance? When a pure tall pea plant cross with dwarf plant in F1 generation, only tall pea plant produce that expression of a one gene in presence of its allele, or expression of one allele in presence of its contrasting allele, is a dominance of that gene, since that small t, another factor, could not express in presence of capital T, is a recessive allele, and the gene which express in F1 generation is called dominant gene, and phenomenon is known as law of dominance. I hope you got it, that phenotypic ratio. And what is genotypic ratio? Genotypic ratio is capital T, small t and small t. Here 1, capital T is 1, capital T small t are 2 and small t 1. Means in his that pea plant crop, out of 1000, near about 500 plant could be with heterozygous condition, about 250 plant could be homozygous condition for tallness, and about 250 plant could be homozygous for dwarfness, and that is genotypic ratio. So that monohybrid cross has phenotypic ratio 3:1 and genotypic ratio. I hope you did understand that monohybrid cross as well as law of dominance. Even if you are in doubt, you can ask in comment section. I appeal you to like and subscribe our channel as well as press bell icon so as to receive you new videos as and when they are uploaded. Thank you.